Hi, all. Welcome back to Environmental Science 101. And we're just going to continue talking about the scientific method. Remember the steps to the scientific method: observation, develop a question, from the question, develop a hypothesis, from the hypothesis, develop an experiment or a test, carry out that experiment test, collect data, analyze the data, and then form a conclusion from that. And that conclusion can be, a publication, a presentation, or conclude that your project's incomplete and that you need to design new hypotheses, new experiments, etc., to carry out that project. We're in the middle right now of talking about Dr. Jenner's work with generating the first vaccination, and that vaccination was for smallpox. And we were talking about his experiment where he would inoculate a child with cowpox first, and then after the child healed from cowpox, he then would repeatedly inoculate the child with smallpox to see if the child was immune or built up an immunity to smallpox. Okay, so remember, the independent variable in this case is the vaccine. The dependent variable is whether or not the child or the patient is protected from smallpox. Okay, and really he didn't have much of a control because he only had a sample size of 1. Okay, so let's look and continue his research. Okay, what he found in his research. So after giving the child cowpox and then inoculating the child multiple times with smallpox, the child survived over 20 inoculations. In fact, the child never died or succumbed to smallpox. So Jenner concluded that indeed an inoculation of cowpox would protect people from smallpox. So that was his conclusion, is that his hypothesis was supported, that in fact cowpox, being infected with cowpox, gives you an immunity to smallpox. Okay. Now, remember, we talked about this before. In science, hypotheses are not proven. Okay? They're just supported or refuted. Okay, so we do not prove hypotheses. So it doesn't prove that Jenner was right. It just supported his hypothesis. His results did. But there's problems with this. Okay. Well, his sample size was one child. He had really no control, and he didn't have any other experimental variables. It was just one 8-year-old boy. So Jenner went out and he tried to publish it. He tried to publish his study in the Royal Society of Medicine, and this is that piece of science that makes science different than a lot of other fields. If you're going to publish your results, you have to publish your results, and they have to be reviewed by people who are specialists or by people who are other scientists. They review it and they may come back and say, no, this is a good start, and that's what they told Jenner, but you need more proof. Your sample size is 1 individual. Okay? And so that's one of the nice things about science, is that it's always checked. It always has kind of a check balance and tries to prevent biases from popping up, or tries to prevent that portion of pseudoscience from sneaking in and getting a publication out of it. But at any rate, often when these studies occur, the study occurs and the general public gets wind of the research, or maybe a newspaper grabs it before, like a journal publishes it. The newspaper might publish early results, and that can cause a lot of issues, and it did in Jenner's case. So Jenner's results was leaked, or his research was leaked out to newspapers, and always, especially in the 1800s, one of the greatest things to do was look at the comics, because the comics were where you're going to see what people thought of your research, or what people thought of your ideas. And Darwin experienced this, Mendel experienced this, like lots of researchers experienced this. Once people got a wind of their research, they would have fun with drawing comics. And you can see sort of here, you can see this depiction of Jenner who is injecting people with cowpox, and then all of a sudden they have cows that are growing out of their arm or their nose or their eye, ear, etc. And that was really kind of a play on his research. But that didn't stop Jenner. Jenner was able to locate several other patients that were willing to volunteer their children. At this time, the information that the 8-year-old boy made it really helped a lot, and lots of parents were willing to donate their children to science. And Jenner even donated his own 11-month-old son to the study. He had enough faith that what he was figuring out would protect his own baby, that he put his own baby in the study. And he got a little bit better at the process of inoculation. He got a little more scientific and refined his experiment to where very few biases were in there, and the same amount of material was given to each individual. And eventually what he was able to do is finally published his results, and that's how the first vaccination was developed. And in fact, vaccination is, the root of it is vaca, which means cow, and that's where vaccination came from, the word came from. Okay? And many of you probably have family members, family members that were in the 60s, maybe born in the kind of 40s, 50s, 60s. From that perspective, they most likely got a vaccine in this form, whether that be the vaccination for smallpox or the vaccine for polio. It was given the same way. And so if you look on either the right or left shoulder, you should see this mark. Okay, sometimes you see two because the individual had two separate vaccines. But the old method of vaccination was to put the postulate on the tip of a needle and then jab it into the shoulder of the individual. And that's how you introduced that vaccine material. And often it would be, the vaccine was for viruses, and often it would be a virus that was not broken up and heat shocked like they currently are. So vaccines currently are injected into you, and the virus has been broken into pieces, so only the protein coats are really left, and the virus can't reproduce. In the case of early vaccines, the virus was for lack of better terms, it was considered alive. Okay, so viruses are not living organisms, but it was considered alive. Therefore, it could infect you. You could get sick from it. And most of the time, like in the case of Jenner's case, he was inoculating or vaccinating people with cowpox to have them fight smallpox. Okay, and so that's why it worked so well. Is he wasn't inoculating individuals with smallpox, okay, but he was inoculating them with a closely or a cousin virus that your immune system would build up a response to that cousin virus, and it provided you protection against smallpox. Okay. All right, so let's talk a little bit more about experimental design. So that's kind of how Jenner went through the scientific method. Okay, so again, he started with an observation, question, hypothesis, experiment, publication. In his case, he had to go back to his experiment and redo it because his first publication was rejected. Okay, so he went back, redid it, collected more data, and then republished. And again, that was, the results of the first vaccine. So experimental design really depends on what is the goal of the research. And, especially in environmental science, controlled experiments they're really not possible. To decide whether or not the planet is warming, or decide what the effects of warming oceans are on coral reefs, or to decide, what the effects of a warming air current on native trees. These things, you can't control those, because you don't have control over the weather patterns like that. You can try to do it in a closed system, in a greenhouse, or in large aquariums, but it's not ideal, and many times it's not possible to look at those effects. So natural experiments are normally conducted under normal circumstances, and we just deal with the fact that you could be out collecting data and it could be raining on you, and then the next time you come out and collect data, it could be snowing, and the next time you come out and collect the data, it could be 40-mile-per-hour winds. And all of these might have the effect on temperature, turbidity, whatever it might be that you might be collecting. In our case, we collect lots of water samples, and if the wind speeds in the region are 50 miles per hour, 60 miles per hour, the turbidity in our water sources can be extremely high because of the natural circumstances. Okay? The advantages of this is it's a realistic study. It's what's really going on. Okay? It's not best case scenario. It's the scenario that happens every day. So if you see that species are fluctuating nesting times due to temperature, and this is a field study, well, that means it's it really occurring. Like, if you can document that indeed they're nesting earlier in the season because temperatures are warmer, then you can see that, you can document that, and that's taking into account the natural circumstances. Like, some days they might start their nests and it's snowing still. Some days they might start their nests and the invertebrates at which they feed their young on they haven't hatched yet. And so these things, these normal circumstances, that's one of the advantages, is that it's more accurate when we're talking about what's happening on our planet. The other piece is it can show an additive effect. So maybe you think it's just temperature that's changing the timing that flowers are breaking petal, or plants are flowering. But really, what if moisture is also involved? What if wind speed is involved? What if the amount of sunlight in a given day is involved in that? Okay, so you can have an additive effect that was never predicted, that wasn't part of your original hypothesis. Okay? There are disadvantages, though, for sure. Is you got to tease out those additive effects. So sometimes you have these additive effects that compile, and you just get a bunch of noise. Like when we look at temperature profiles of different places on the planet, you will see a bunch of spikes and noise around the temperature, and sometimes it's hard to decide, well, is this region actually warming, or is this kind of a natural cycle that happens in this region over and over again? So sometimes it's hard to develop that meaningful control or that meaningful ideology when looking at the data, because there's so much noise around it. Okay? And one of the key components, and we'll talk a lot more about this as we progress, is when you're in normal circumstances, it's hard to remove the biases. Okay? If you're a field researcher and you're like Jane Goodall sitting out here watching chimpanzees, and you're just getting drenched in rain, it's just pouring down on you. It's hard to want to be out there for the entire period. Or if you're sitting there and the mosquitoes are so bad that you're finding it hard to concentrate on your subject matter because you're just getting consumed by mosquitoes. These are biases, okay? One day, if you cut your experiment short by 30 minutes, that's a bias. If where you sit down is a bias. What group of organisms you're looking at is a bias. And so there's lots and lots of ways at which we have a very difficult time in natural experiments to remove all those biases. Okay? And, similar things when we talk about climate change. We're drawn to the extremes. Okay? As a researcher, we see something like this, we take a picture of it, that's going on the cover, or that's the picture that we have in our mind when we talk about climate change. We talk about, polar bears, and running out of ice and having no place to feed. When, if we look mainland, there might be 10, 15, 20 polar bears that are on the mainland that are just roaming the mainland and consuming material. But we're drawn to this one that's, hanging on to the last sheet of ice. It's a bias. Now, does it mean that the natural experiment, does it mean that your experiment is faulty? No, but it does show that you are drawn to an extreme, and it kind of points at this inability to be a neutral party. Okay? And that's where the scientific method comes in. The scientific method, it's specific, and there's specific steps, and each one of those steps needs to be followed, and that publication step is really important, because in that publication, if another researcher looks at your data and says, sure seems like you're just choosing polar bears that are sitting on little sheets of ice to do research on, what about all the other polar bears? What about the regions where the ice melt is not as drastic, or these kind of things? And so those biases are difficult to remove in natural experiments, but the natural experiments is where we get a lot of our data, especially in environmental science, because there are so many systems that are connected, whether that be how much water, temperature, the wind speed, the effects of humidity, and the effects of how much sunlight a region gets, and all these things are additive and important for predicting things that might be occurring in a natural system. Okay, next time we come back, we'll talk about margins of error, and then I'll give you some more examples of some biases that occur in research next time.